All right, so good afternoon, everyone, and welcome to RSC's Introduction to RSC Technology Supported webinar series. So today, we have Atlantis Bioscience here with us today. So I'm Jocelyn, I'm from RSC. So in the meantime, before we start the webinar, do have a look at the webinar tips, all right? So if you have any questions, feel free to click raise your hand icon if you wish to speak up, or write them in the Q&A section by clicking on the Q&A button below in the control panel. So let me see. All right, okay. So now that we have started the webinar, let me introduce Yvonne. She's from the Atlantis Bioscience team to share more about Atlantis Bioscience today. All right.
Right now, here's my share. Hi everyone, can you see my slide? Okay, so hi everyone, my name is Yvonne. I'm from Atlantis Bioscience. So before we begin the presentation, I would like to briefly introduce Atlantis Bioscience. Atlantis Bioscience is established in 2016. We are a private company providing life science products and services catering to various needs of biomedical and healthcare research in Singapore and Asia Pacific. So our interests are mainly towards the translational research, such as those in oncology, cell and gene therapy, for example. So we have a wide portfolio of services and products, and here are some that I would like to share with you. So this includes the basic cell culture reagents, such as the cell culture media, animal serum, imaging dyes for cellular stains, Western blotting and flow cytometry reagents. In terms of oncology and drug development and delivery, we have hydrogel-coated labwares that can provide you with different stiffness for your cells to mimic the tissue environment. We have human-derived ECM and the spherical plate for 3D culture, so for those looking into organoids or spheroids formation. Primary human liver cells for the study of drug metabolism. Nanoparticles, MSC-derived exosomes, human serum albumin for drug delivery. So in terms of services, we have the preclinical oncology services, which include the in vitro and in vivo platform. For in vivo platform, we have the animals models such as the PDX, CDX model, humanized models, and the syngeneic mouse models. For cell and gene therapy, we provide specialized serum-free media for NK cells, T cells, and MSC. We also have human platelet lysates and the GMP growth factors for clinical applications. Okay, so we are also moving into bioprocessing, where we offer cell factories, microcarriers, and plant-based growth factors. We also provide DNA reading, writing, and editing services. So if you are looking into gene synthesis, this is something that you can write to us as well. So as we are now moving into a more digitalized world, we are looking to help move into digitalization with the use of electronic lab notebooks. So something you may want to consider, especially if you are from a startup or in industries, if you want to have a proper project data or inventory management. So all in all, Atlantis Bioscience, we are a one-stop platform able to provide you with different tools and solutions to help you to complete your research workflow. So if you have any inquiries, please feel free to write into Atlantis Bioscience. Do visit our website and also our social media platforms for more updates and news. So now, before we move on, I would like to invite you to do a quick poll. So we have 3 questions for this poll. Would appreciate if you can take a minute or so to complete the poll. And I will share the poll result with you later on during the Q&A section. So this is the poll. Okay, I think probably we can. Jocelyn, is the poll okay?
I think we can give them some more time to do.
Okay, sure.
So do share with us. Okay, we'll leave the poll on for another 20 seconds, then I will end the poll, all right?
Okay.
You may proceed, Yvonne.
Okay, thank you. So let's move on to today's webinar. Correct. So in today's webinar, we have invited one of our service providers whom we have been working very closely with, Crown Bioscience, to give a presentation on using Tumour Organoids platform for cancer drug discovery. So Crown Bioscience is a leading provider for oncology services and preclinical cancer models for drug development. Dr. Xu Xiaoxi is the speaker for today's webinar. She is currently the director at Crown Bioscience in charge of all in vitro teams in the China side. Received her PhD from the Chinese Academy of Sciences. She joined Crown Bioscience in 2015 and has studied the key hub IP protected organoid technologies from the Netherlands. And this is what she will be sharing with us today. So now let me hand over to Dr. Xu.
Okay, thank you. I'll start my presentation. Okay, in a minute. Okay. Can you hear me all right?
Yes, we can.
Thank you. So today I'm very happy to join this session and have the opportunity to introduce our biobank to you. So my title is a living biobank of matched patient-derived cancer models from both in vitro and in vivo for cancer pharmacology. Okay, so before that, a little bit of the background introduction. So as you might know that for the drug development, the translational efficacy is quite low. For example, it shows that even in the clinical phase, even from phase 1 to approval, the drug approval is, the percentage of the drug approval is quite low. For the non-oncology drugs, it's only 12%, and for the oncology, 5%. And with all this translation, each drug would averagely use 15 years and 1 billion. So that would mean, so currently what we have done for the preclinical side is not so perfect to do the clinical to do the clinical evaluation. So that caused the gap between the lab work to the web work. So that's by a lot of reasons, and one of the main reasons is that the human cancers are very complex. As we know that only derived from different tissue, which shows the histopathology, but is also can be characterized by these 5 key parameters. For example, like molecular pathology, because by the evolution of the technology of screening, like RNA-seq or WES, this kind of molecular pathology has taken very much research on that, using to develop targeted therapy. And also by killing most of the cancer is still not done. It's cancer stem cell driven, which shows the tumor heterogeneity, which would also cause drug resistance as well. And most complex part is that it has a tumor microenvironment, which consists of not only the tumor cells, but also the stromal cells and immune cells, and also all the cytokines pathways to work together to make the whole cancer. And in all, that means we need to model this complex model at most in the preclinical phase, so that we can have a more relative drug efficacy there to predict the clinical effect. So for years, it's now being accepted by the market that the PDX model, which is patient-derived xenograft, this kind of xenograft were using the patient tissues directly inoculating to the mice and generate this kind of complex model, is now the workhorse for drug predictivity. For example, like since 2019, FDA has approved 43 drugs, and like 17 of them were novel oncology drug approvals, and of that 17 drugs, Crown has directly contributed to 7 of them, and 6 of them were assessed by the PDX models. So that means the PDX models are quite predictive, and you can always predict the current patient if efficacy using this kind of model. And it has all these key parameters which mimic the patient sample as well. And on the other hand, the US Cancer Institute has stopped using the cell lines to do this kind of drug efficacy prediction. So for using this kind of PDX biobank, we have already generated over 3,000 models, which has very large diversity from like patients are from US, European, and Asians, and also from different tissue origin, from blood cell to solid tumor, and it's like the most variety of the cancer collection. And using those models, you can easily test your drug efficacy, and then you can move this drug evaluation to the clinical phase. And for those parameters, we have done so many data sets for you to choose from. We have a comprehensive online database that you can search for each model's characterizations. And we also have done like many clinical trials using the mouse. And for each model, we all collected those RNA-seq, both RNA-seq and the WES for the gene sequencing, and you can find gene expression and mutation information in that online database. So it's very easy and friendly use if you want to do such a PDX study. But using PDX model is not enough. It is an animal model which have the apparent shortcome, which you cannot do a large screening using the animal model. And also for the animal model, it's quite complex. If you want to do the drug mechanism of action investigation, then there's some problem. It is better to have a corresponding in vitro model that you can also work on. So by this being said, well, the organoids are the most perfect model for such needs. Well, like 10 years ago, Tumour organoids has been brought up into the research, and it is a very new method that directly use patient sample and can convert it into cell culture. So this kind of organoids can continue growing within the lab, and it also consists of all the tissue features using our current media components. So it has most of these key parameters, not including the tumor microenvironment, because currently only the tumor pads can grow within the organoids. But the good thing is that for organoids growth, it can not only keep expansion, but it can also maintain the morphology, the genetic feature, and also it can be cryopreserved so that you can have a big biobank and do this repetitive study using it to see whether the drug efficacy, and also you can use it for research as well. Here on the right side, you can see a very typical culture of the organoids. This is one of our model in our biobank. It has this cystic look, and it has a hollow inside. We have passages for more than 30 passages. It remains the same. And also we have tested the drugs and also the sequencing results. Well, this organoid model can maintain most very good morphology and characterization as well. And for if you look at in live cell, it shows like this hollow balloon. But if you cut it open, here it is HE staining of the organoids. You can see the cells aligned together in a very peculiar way. And this structure can be also seen within the tissue. And in the below image is a tumor section, shows the HE staining as well. So this structure are quite similar, and this is automatically formed. So this organoid has a much better similarity to the in vivo side. And in addition, we also use IHC to see the biomarkers expression. So here on the left side is the tumor biomarker staining, and on the right side is the organoid staining. And by these cancer stem cell markers we have stained, you can see the percentage distribution are similar. Shows that only the organoids has this heterogeneity, but also it has the resemblance of the in vivo tumor. And using organoid for drug prediction is quite popular now. For example, for this science publication, which happened in 2018, which is the first publication using organoid to predict the clinical drug effect as well. In this paper, there are positive prediction and 100 negative prediction. So this means that using organoid as the in vitro screening model, you can over 90% you can predict the patient clinical reaction. So that paper has like dropped a bomb in this research area, and this is not the only one, but also in later, like in 2019 and 2020, there are like other similar study designs, but on different drug treatment and different group of patients as well. They all show very similar results. Well, overall, it has the over 80% predictive, which is never seen before. So that means using organoids for drug prediction is quite hot now. So that would make a like circle from you, because you use the same patient, can also generate a PDX model, which is the in vivo model. You can also generate a PDO model, which is the corresponding in vitro model. So that makes a perfect translational chain. So by this concept, Crown has also developed our corresponding biobanks. For example, when we use we have the patient tumor, we either directly using that to generate the patient-derived organoid, which is called PDO. We can also use this PDO to inoculate into animal to make a PDO-X. And in the meantime, we can also use this patient tissue to generate a PDX first, and then extracting the tumor, we can also use this tumor to generate the PDXO as well. So that means we have a very comprehensive and a comparable in vitro and in vivo biobank. So for the organoid, currently we have over 500 models, all from different cancer types. Currently, we have 16 cancer types available. And for organoid model, what's more special is that it has corresponding normal counterpart, so that you can design a tumor normal pair reaction for your drug as well. And we also build a organoid database, which is called Organoid base. This online database can be registered and open to everyone. And we have incorporated all the patient information and also morphology, histopathology, and also the RNA and WAS data in our online database, so that you can have a direct search to find your preferred model there. And we have some models that we have the center care to give you a concept whether you want to do the combination as well. And we didn't use the tumor to do the RNA sequencing. We used the organoids to do the RNA sequencing directly, so that we can compare both the RNA-seq results between the tumor and the organoid. So using organoids for drug efficacy evaluation is quite simple. It's similar to the cell lines. For example, we need to expand this organoids, and then we plate the organoids and then treat the organoids. Then we have the cell viability results. And it's also very flexible and easy to do the combination screening. And by this kind of combination, we can use the matrix design, which allow like drug A in vertical diluted and drug B in horizontally diluted, so that you can have matrix results. And then we can use our Crown developed trouncing method to give you a very direct results to show which where the drug have synergy effect as well. One note to do the organoid screening is that organoids grow quite similar to the in vivo, so that it has longer expansion for passages. For example, the average passage time for organoids is usually 7 days, and also the split ratio would be like 1 to 2. So that would have a relatively longer expansion phase, but we have all characterized all of our organoid models. We can provide you a timeline for each model as well. And for during the screening, because we are using the whole organoids to screen, it is quite challenging to have a low CV value. So for example, our standard template is 384 well plate, and then we use triplicates, and our quality control value would be Zim prime factor, and we need that to be over 0.5, so that we have a solid drug screening plate. By doing that, we use this machine-involved procedure to make sure that our organoid screening is valid. And not only we can test the cell viability using organoids, but we can also use all kinds of other readouts. For example, like biomarker, the downstream biomarker analysis, which can be used for flow cytometry, ELISA, Western blot, and qPCR as well. And we also have a HiCon imaging readouts at our Netherlands sites. So using this HiCon imaging technology is quite evolutionary for organoids, because it has this confocal part which shows the inner structure of the organoids in well. So we also use really 4 well plates for this high-count imaging results. Here is a picture showing what this readout can give us. We can, for example, like in the endpoint reading, we can use staining to stain different parts of the organoids so that you can have a different biomarker analysis and also for the organoid alone, the morphology like area and the roundness can also be evaluated using only 1 well, so that you can have so many levels of the results to rely on. For example, like there's some drugs that don't kill the cells, but they can affect the morphology of the cells, then the high-count imaging would be best for you. So using our using Crown Bio's biobank, you can see these different models for your selection. For example, from one patient, you can have the corresponding PDX and the organoids, and also the PDXO, which derive from the PDX as well. So well, because we have all the NGS results for each model, and we have them for both organoids and the PDX, so that we can have a direct comparison. For example, like when we use the large data sets for the correlation, the first one is the mutation concordance, the mutation correlation, to see whether the organoids and the PDX tumor has the same mutations. When we derived those mutations and compare them together, each dots here shows 1 pair of organoids and PDX, and this percentage means where the mutation number that are seen together. So compare with PDX organoids has averagely the 98% consistency with the tumor. So it's very high, it's almost the same. And then on the other hand, with the MR expression analysis by the RNA-seq, we have 95% as the average correlation. So that means this percentage of genes express similar within both in organoids and PDX. And using PDXO, the PDX-derived organoids, we can also generate the corresponding xenografts. And you can see here that in a regrowth, this organoids in tumor is the growth rate is quite similar to what we have done in the PDX, and also the drag effect is also similar as well. And what we have concerned most is whether this organoid drug efficacy has good predictivity with the in vivo tumor, and the answer will be yes. By our in-house data, the overall prediction is 86%. And by the method, we use the IC50 to show the in vitro drug efficacy, and we use IC50 to set a cutoff value to define sensitive and resistance. And then we use TGI for in vivo efficacy to define sensitive and resistant. This is the same method that has been published in the previous science paper. And you can see here, if you see more closely, the positive prediction is 75%, and the negative prediction is 91%. It's quite high, and it's higher than any we have done for the previous in vitro work. So that's very impressive. And using those big data, we have done more analysis. We have included more. For example, we not only have the organoids, the PDX, and also we have some patient-derived cell lines data. we combine those together, and then we use public database, which is TCGA, which consists of the patient sample, and also the CCLE, which consists of the current commercial available cell line samples. So we combine those samples together, and in different cancer types, we use that to do we use that to do more. So here shows some results. So as the overall PC analysis, you can see here very clearly in this figure that, well, the TCGA samples are aligned here, and the CCLE samples are aligned here. And then in between would be the PDOs and PDX. So that means this kind of in vitro model and in vivo model did fill the gap between this between the patient and the cell lines. And on different on another PC analysis, it also shows very similar results. And if we look at by different cancer types, for example, like the colorectal, lung, and pancreatic, we can see similar trends. For example, like the PDOs, then PDX, and then PDXOs, then PDCs, then CCLEs. So that shows very similar trends in either cancer types we have tested. And for paired data sets, we have been done for pathway analysis. You can see that for in organoids, it has lowered EMT activity and reduced extracellular matrix related expression. And this is quite, and this fits our hypothesis because organoids currently growing in vitro, it only consists of the tumor part. It doesn't have this tumor microenvironment and all the extracellular part to support. So it lacks this kind of activity. But organoids is a in vitro model, but it has this kind of flexibility that you can add those parts in to do the co-culture as well. And for other pathway analysis, you can see that compare with the PDX, like the oxidative phosphorylation, lipid metabolism, translation, and transcriptive also upregulated in the organoids. Because in a in vitro culture environment, the nutrients and also the oxygen are quite sufficient. So, so the cells proliferate a little bit higher in this in vitro culture. So it's also good to see that this findings corresponds with our hypothesis. And for the down-regulated pathways, you can find that like inflammations and EMT and those related pathways like angiogenesis. So that pathways are down-regulated in the organoids because like organoids lack of that part. So it's also good to know that. And for different cancer types, you can see here, well, similar results can be shown in different cancer types. Also, like inflammation, EMT are more active in PDX tumors, while this metabolism, translation, and transcriptive are more active in organoids. And here's example of like HIF-1 alpha signaling pathway. So like if you want to investigate this signaling pathway in organoid, currently it's but we can use this organoid in a low oxygen culture environment, because for example we can set our incubator to be like very low oxygen percentage, 1% and all, and then it can also activate this signaling pathway so that we can have this more MOA investigation. So to sum up, we have built this patient-derived biobanks, consists of both in vitro and in vivo, and they can be very clinically predictive. And you can use it to test your hypothesis and validate your drugs. And for using organoids alone, it can be quite flexible. You can use it for combo screening, and you can have a very direct drug synergy effect to show. And then for this mesh biobanks, you have a better translational effective, and it's very good for the drug development. And also currently using organoids is not very perfect. It's can still develop the epithelial related cancers. For example, like sarcomas or lymphomas cannot be generated into organoids now, but still the scientists are finding ways to generate organoids for that aspect as well. So currently we can deal with the solid tumor first. So if you have any requests regarding this kind of models, and please contact Crown Bioscience. And thank you again for your listening, and I'm happy to take any questions.
Yeah. Okay, thank you, Dr. Xu. So before we go into the Q&A session, so can we take a look at the poll results? So in the meanwhile, for those who have questions for Dr. Xu, you may formulate your questions and type it in the Q&A chat box that you can find at the bottom of the screen. So Justin, can you share the result of the poll?
Done. It's shared.
Oh, okay. So basically, for the first question that we asked was about the what stage of drug discovery and development are you at? So it looks like a lot are still under target identification or the preclinical testing stage. So for the next question was, what sort of platforms or model systems would you use for your drug discovery process? It looks like a lot are leaning towards using of Tumour organoid, which is really good. That's what Dr. Xu has shared. And many are also towards the humanized mouse models and the PDX, CDX models. So just to share, so Crown Bioscience also offer platforms for the PDX, CDX model and humanized mouse models. So if you're interested, you can write into us to ask for more information. Yeah, and the last question was mainly about, just the top 3 techniques that you use in your laboratory. So mainly was cell culture, flow cytometry, and the animal work. Okay, so maybe let's move on to the Q&A. So do we have any questions for Dr. Xu? So you can type in your questions here in the Q&A box. So maybe let me ask a question for Dr. Xu. So can all these organoids be used for toxicology studies?
Usually we use normal organoids for toxicology. Currently the Crown, we have this license between Crown and the Hub. The Hub is the institute which have developed organoid technology. It has the patents and it gives license to commercial companies like us. Currently we have this preclinical oncology license, but not have the toxicology yet. We can use organoids to test the toxicology, but Crown cannot yet due to the license issue. Yeah.
Okay, I see. So how about, can these organoids be engineered?
Yes. We have already have like dozens of organoid lines being engineered in applications like if you want to label with GFP or luciferase, then it's very easy and relatively easy. And you can also use organoids for gene manipulation, like knock-in, knock-out, so that and overexpression, so that would be no problem.
I see.
It's the difficulty is harder than the cell lines, but it can be done.
Okay, so do we have any questions from the floor? Oh, I think we have one. So this question is posted by Pei Yen, asking, does the PDO's omics change from passage 1 to passage 30?
Yeah, well, yeah, this is a very good question. We have tested over 20 lines, and the passage gap would be over 15 passages. And well, using RNA-seq as a method, the result would be yes, that the consistency of the organoids in vitro culture would be very good. For example, like the organoids in culture, well, we for the method for the passage is that we disruptive the structure, like mechanically, it will be like into the fragments. Then on the next day or in the next 2 hours, you can see that each fragment would form a very round organoid structure there. So it's happens automatically, and this kind of structure is quite strong. So I think by using this, the gene expression and also the stability of the genome are quite steady. Yeah.
Okay, so we have the next question from Carl Tan. How different are PDXO and PDO in terms of transcriptome, drug response, etc.? And the second part is the availability of PDXO versus PDO just a matter of how successful either culture is?
Yeah. Well, currently, in terms of like the origin, we developed the PDXO because we have a large set of the PDX. That also means a patient library, and we can also directly made PDOs from patient sample. But unfortunately, currently, it is hypothetically available. We don't have any PDXO and PDO from the same patient yet. So, well, due to our current findings, the PDX and the organoids are very good at the stability of the transcriptome. I think the answer would be also quite similar. And also for the second question, well, for due to our experience, PDOs, the successful rate for PDOs are higher than PDXOs, because because PDX has already well, the PDX tumor alone already has a 20% successful rate, and we're using PDX tumor to generate the organoids, which would affect the results will be the mouse content. So the mouse content can sometimes overgrow in the organoid culture, and we monitor that very closely. So that would cause the successful rate of generating a PDXO would be lower than generating a PDO models. Yeah.
Okay, yeah, thank you. So we have one question coming from chat earlier. That was from Xueling Guang about nice presentation. Do the PDOs recapitulate the heterogeneity in the patient tumor?
Well, yeah, that's the concept of the organoids. So the organoids are derived from the adult stem cell, and also in the cancers, we assume that the organoids are derived from the cancer stem cell. So this kind of proliferate, not only proliferate, it can also differentiate as well. So it's a mixture of different populations of the tumor cells. So it shows very good heterogeneity. And this part is quite similar to the corresponding patient tissues. Yeah.
Okay, thank you. So there's one from Gopu. So this is about head and neck is the complex region that includes tumors involving eye, nose, throat, mouth, and salivary glands. Do you have information related to models for oral cancer?
Well, yeah, but not much. I think we have less than 3 head and neck model available. But I have to check our list and get back to you later. Or you can log into our website to find out more about those models, or contact our Atlantis. Yeah, we can send you the list with the patient sample.
Okay. Yeah, thank you. So any other questions? So actually, I would like to know about whether if we could use these organoids for immunology studies, like co-culture with the immune cells. Would that be possible?
Yeah, well, using organoids in co-culture is actually one of our routine assays now. We use the, although we cannot have the corresponding patient immune cells, we can use the healthy donors, PBMCs, and co-culture them with the organoid to see the tumor killing effect. We can use this culture system to evaluate well, the T cell killing effect, and also the immune checkpointer. And also we have done like CAR T cell killing and bispecific and antibody evaluations and so on. So yeah, that's a very good.
Okay, thank you. Is there one more question? Oh, there's one more question about is that how can we procure? Is that organoids? Is that a spelling error? Do we need IRB approval? Is that organoids? Because it's spelled as TYE. So I assume that this is a spelling error.
Well,
what do,
well, if it is from epithelial, if it is from epithelial, it can be used to generate organoids, but still we need to.
Dr. Shi is the typo, so basically it's asking how can we procure these organoids? Do we need IRB approval?
Okay. You mean like to buy the organoids from Crown? Well, unfortunately, due to the license from Hub and Crown, we cannot sell the organoids, but we can use organoids for service. But no not live cells can be bought, but we can send you the like FIPs or cell pilots if you want to do some in-house analysis using those samples first. But for the like cell viability evaluation or like the like in vivo modeling, you have to do it within Crown. Yeah.
Okay, so this one we cannot, Crown do not sell the organoids, yeah. Okay, thanks. So do we have any further questions for Dr. Shi? So if you do not have further questions for Dr. Shi, and if you have questions later on, you can send the questions to me. So I can send it to Dr. Shi, and we will address that later on. So thank you, Dr. Shi, for your time. And before we end this webinar, I would like to hand it over to Jocelyn, because I think she will be sharing with you a post-webinar survey.
So thank you, Dr. Shi and Yvonne, for the wonderful presentation today. So dear attendees, before you leave, please do our post-webinar feedback form. Do give me a moment, let me share. Okay. So we appreciate your feedback so that to help us to plan for future webinars. So let us know if you have any content you would like us to present to you, to bring to you. All right. So do complete this short survey. It's just a very short 2-3 minutes survey to complete to do. All right.